Okay, and so now we're live with the G Plus Micro JC, the Google Plus Microbiology Journal Club. And so after some technical difficulties that have been somewhat resolved, I think we're still missing one person who hopefully can join us if all the technical problems get taken care of. But we have a nice group here to talk about the study that was just published recently, Geospatial Resolution of Human and Bacterial Diversity with City-Scale Metagenomics. And so before we get started, I thought maybe it would be good for us to introduce ourselves. So my name is Laura Williams, and I'm a new assistant professor at Providence College in Providence, Rhode Island. And I'm actually teaching a course right now for non-majors on microbiology. And this study was really well timed because we just finished our microbiome module. And we did a class on the microbiology of the built environment. And one of my students actually two of my students sent me links to this paper that they saw in like things that they were reading, like the Wall Street Journal. So it was really cool that it came out right at a time that we were talking about topics that had to do with this type of study. So I was happy about that. And with that, I will turn it over to the rest of our group to introduce themselves. Maybe we'll just go in the order of the panels. So Mark, you're right next to me. Do you want to go next?
Hi, I'm Mark Martin. I'm an old associate professor at the University of Puget Sound in Tacoma, Washington, and I'm delighted to be here to participate. Did you want me to say anything else?
You can if you want to.
Well, I've become very interested in the interactions between microbes and humans, and microbes and, in fact, all other organisms, and so this is a wonderful example of the cloud in which we live. And I'm looking forward to this. I wonder how, and this is where I think Dave is going to be very helpful to us, it's possible to have what I call swab stories, where you find here's some terrible microbe that we found in any given environment, and then the press runs away with that. So I'm very interested to see the sober, cold, serious approaches that we're going to put on this.
That's great. Karen, do you want to go next? Oh, you're muted, Karen. You have to unmute yourself. There you go.
Sorry about that. I'm Karen. I'm a PhD student at Simon Fraser University, and I study a bacteria called Francisella tularensis, which is a potential bioterrorism agent. So in the context of this paper, this is quite fascinating in the sense that you can find these that people claim that they can find these things in the natural environment, which we'll talk about. And well, but that's it for me.
Okay, thank you, Karen. Juan, do you want to go next?
Right. Everyone can hear me okay?
Yep, I think we've got you.
Well, so my name is Juan Ugalde. I'm an assistant professor here at the Universidad Mayor in Santiago, Chile. So I'm providing some international approach to this discussion group. Also, I'm very interested in this paper, and that's why I wanted to join the discussion today, because my background, my training was in microbial ecology, microbial diversity, and I think it's very interesting to also start looking for microbes, not like in extreme environments, like you go elsewhere, like the deep ocean or other places, but also, as Mark was saying, what's around us, so the cloud of microbes that we are living within. So I think this is a very interesting paper for discussion, and I'm looking forward to what we are going to be talking about today.
Great. Thank you very much for joining us. We're really excited to have people from outside the U.S. That's great. So if Willem gets here, you'll be a party of two people from representing international places. So and last but not least, we have two people joining us in one window. One might have a little more to say than the other, depending.
Oh, I think that's going to change. She has a lot to say. She just has a limited vocabulary. Dave Baltris, University of Arizona, and 17-month-old Hayden Baltris, who misses her mommy. So we'll see how that goes. But interested in everything.
We're sorry that she was used to Skyping with her mom, and we got on the Skype, and it's like, oh, that's not my mom. I'm sorry. We're going to talk about microbiology.
All good.
Great. So thanks everybody for being here and being patient through my struggles with Google Hangouts. So we got it worked out, hopefully with a minimum of hassle. But as Juan said, I think this is a really interesting paper to start to tackle the issues of the microbiology of the built environments around us. And I know there are a number of groups that are focused on this specifically. There's a research institute at the University of Oregon where they're specifically doing work on the microbiology of the built environment, trying to understand how microbes colonize and persist in man-made environments. And since we spend over 90% of our time indoors in our lives, it really makes sense to take a look at the microbiology of the world around us, our offices and classrooms and buildings. This is a particularly interesting example of that because it's in the New York City subway system. So these researchers wanted to understand the microbiology of a built environment that was highly trafficked. So it has a high density of people coming through it. It's in a metropolitan area of New York City, which is one of the, I think they said it's the 8.2 million people live on a landmass of only 469 square miles. So this is a lot of people moving through this particular system. And so there's a lot here, and I thought it kind of broke down into 3 general points that occurred to me. One would be kind of what did they find specifically that they're drawing these conclusions, and another would be kind of what got focused on by the media that maybe isn't as well supported, and then the other point I think is just kind of a methodology point, like how do we see these kinds of studies moving forward. So there's a lot of angles that we could go at to tackle this, but I thought that maybe we would kind of start with the methodology because I think that sets a framework for interpreting a lot of the results. So they did a shotgun sequencing analysis, so it's a metagenomics study. They swabbed a lot of surfaces, and it was this was a pretty tremendous undertaking. So they took all of these samples from different stations within the subway system and all the subway lines. And I think in the end they had, how many samples? 1,457 samples, which is pretty extraordinary. And then they did Illumina sequencing to try to get DNA information about what was present. So if we look at kind of the pie chart that they've got in Figure 1, one of the main things they emphasized was that almost 50% of their sequence reads came from an unknown organism. And then another 47% came from bacteria. And so I was really curious about, given the methodology, how do we feel about how accurately we can classify down to like the species level with what they've got grouped into the bacterial part of the pie chart. So I thought I would kind of put that out for people to have comments on the methodology specifically.
The first, I don't have the link. Can you guys hear me? Or is that...
Yes.
I don't have the link, but you can fish up Nick Lohman's. Basically, Nick took E. coli K12 and split it up into 100 base pair reads and just ran it through the analysis. Yeah, and it comes up. It
looks like this.
Yeah, there's a lot of unclassified stuff there, right?
Yeah.
So that's the first, right? There's just not a lot of information, or there's not as much information in 100 base pair, 150 base pair reads as you would like to be able to truly classify things. So I bet there's unclassified stuff out there, but I'm not sure what percentage it truly is of everything. Yeah.
Yeah, so I mean, you went right. I went ahead and printed this out because I felt like it was really helpful. So you went right to it. So here's what Dave was talking about. So Nick Lohman posted this on Twitter as an example of what happens when you try to do metagenomics with short reads. So what he did was he took K12 E. coli reference genome and he shredded it into 100 base pair reads, and then he sent it through like a metagenomics pipeline. And what you can see is that you get, a lot of what you would expect from being, Escherichia, and you get some Shigella, but then you get a lot of stuff that it definitely is not. So I think Dave's right. That's something that we have to be cautious about in terms of classifying such short reads.
I think And also, one of the main limitations is basically that you can only classify what you already have on databases. So if you're looking at basically that 50% of unknown organisms are probably things that maybe are not so novel, but they're not on the databases. So you don't have anything to compare with. You don't have anything to classify.
Mm-hmm. Yeah, and you're right. They made that point with like the representation of eukaryotic samples in the database. I think they said that this was a point where they had difficulty assigning reads because there's just not enough complete genomes for eukaryotes in the database.
But my guess is that probably also besides eukaryotes, I think that's probably true for viruses as well. I wouldn't be surprised if a lot of the sequences that they found on the study are just novel pages that basically you don't have anything to compare with.
Yeah, that's a great point.
So the second point I want to make about the methodology is basically bacteria are relatively easy to get DNA out of with the mobile kits, but their eukaryotes and especially things like fungi are a lot more difficult, and a lot of the fungi are kind of recalcitrant to actually isolating DNA, and so that's probably another reason why they miss. You're going to miss a lot, but there's going to be some DNA prep biases here too, and that's why I think they're getting back most of the. There's definitely fungi out there, and they're just not represented in this data set. So I think that's one of another main issue.
Do you think so? I wondered if I understood from the methods right. I think they were doing alignments against the nucleotide database for NCBI. And I can't recall off the top of my head. Does anybody know, does that include, that doesn't include metagenomics studies, does it? It just includes, so I'm curious if they tried to align them against other metagenomic studies, would they have found more useful information, or would that have just confused things?
So I think it's, if they used the one that I'm thinking of, it's only genome sequences and sequences that have been uploaded to NCBI, but it doesn't include any environmental sequences. So
right, that's what I wondered.
Yeah, so I'm pretty sure that it doesn't include any metagenomic or any environmental sequence, any metagenomic data.
That's what I thought too, and it made me wonder if they had done a separate analysis where they had taken their reads and tried to see for other similar studies how well their reads kind of map against those, just to get some sense of, is this like a typical sample? Because that unknown organisms portion of their results is so huge. It just made me wonder if there was any way for them to get a sense of what are the reads in there, and do we see them distributed in other types of metagenomic studies?
At least on a few of the metagenomes that I've been working on, we usually see around maybe 35–40% of things that we can really classify, at least on the first pass. Like when you do something like this, just compare everything against NCBI database, for example.
Mm-hmm.
So sometimes you can improve that if you start doing some assembly or start using some other things like nucleotide properties of the nucleotide sequences. But usually, the 48% number, it's a little high, but it's not that high, at least for me, for what I'm seeing on other datasets.
Right, and they did make the point that there was a study of microbes in the air, I think, in New York City, where they found a similar proportion of reads that they were unable to assign or classify. So yeah, that's true. That's a good point. That we've got a lot out there that we're recovering that we don't quite know where to put it, or how to classify it.
I tend to think of this as drinking from the fire hose, and it has the problem of doing so, and I've seen it in some research that we've done. We have full-length 16S sequences versus short reads, and you get a whole bunch of reads from the short, and if you look at the phylogenetic assignments you get, you don't get the detail that you're used to by looking at full-length, but you have a great deal more depth of what you're looking at. And I'm very concerned, as Dave has brought up, with what the aerosol brought this to mind. In aerosol, you're going to get lots and lots of spores, and I have no idea how well they open up with the power soil kits. Maybe it's all fine, but there's going to be bias no matter what you do, and I don't see a way around that, honestly.
Yeah, that's a reasonable point. Whether or not, there's going to be some recalcitrant there's going to be some difficulties in recovering a representative sample just because of, as a couple people have pointed out, just the methodology of the kit itself might be enriching for particular organisms.
Mm-hmm.
From a bioinformatics perspective, when I looked at the methods, it seemed like they took their reads through a so first of all, they have 100 base pair reads. I thought it was 125 base pair paired in reads, but I think looking through the methods that it's actually 100 base pair paired in reads. And then one of the first things they did was they trimmed the first and last 10 base pairs off. So at this stage, they're basically dealing with 80 base pair reads. And as I was reading through it, it looks like they said that in their megablast pipeline, they only used one pair. So unless I'm reading this wrong, I was kind of thinking, wow, like at that point, you're basically going off an 80 base pair sequence.
And I think this takes you to Nick Lohman's point.
Yeah.
And very directly. I don't know if this is possible because I'm not a computer jock, but if you took, say, the green genes 16S, and you decided to just look at 80 bases and use that information and resubmit, I wonder what the differences would look like. And my guess is you get a whole bunch of unknown stuff that really aren't unknown, you just don't have enough of the sequence to identify it. And maybe I'm speaking out of turn. Dave knows, I mean, he and I have talked about this before. What do you think, Dave? Maybe he can't hear me.
Oh, he's muted right now.
You just lose information, right? So especially depending on which parts you cut out and what, you just lose information with the short reads, and then it's all of the alignment and the matches are based on matching, right? And so the less information to match, the worse your ability is to see things, just because you get a lot more false positives.
So I'm just worried, Dave, if you took a collection of things that were really well known, again, like the green genes set, and you just took 80 bases of each of those, and then what would it look like comparatively? And my guess is you get not just blur. My guess you'd get a lot of unknown things. Now that's something that folks like you and again maybe Nick Lohman could do. And again, I'm not putting down the huge amount of work that was done in this paper. I think it's a trade-off versus, depth versus breadth, for lack of a better term. What do you think, Dave?
Yeah, I mean, you're exactly right. Especially with the 16S though, the 16S will give you the 16S is a lot better characterized, and so you're gonna basically what's gonna happen is if you shorten those reads, you're getting a lot more false matches to something it's not, rather than unclassified.
Got it.
With the genomic stuff, you're going to get, that's where the unclassified part comes in because you're not sampling. The 16S is a lot better sampled than everything metagenomically. And so the shorter the matches, the less chance you're going to actually hit something that's related in the metagenomic database. Versus the 16S, where the shorter the matches, the more the chance there is to hit something that's not it, if that makes sense.
Yeah. Oh, hey, I just saw Willem.
Hi there.
Are you here?
Yeah, I am. Can you hear me all right?
Yes, we can hear you just fine.
Okay, great. It's been a nightmare.
Oh no. Well, we had some technical difficulties on our end, and then it looked like maybe you were popping in and out. So we're glad that you've made it.
Yeah. Okay, glad to join. I followed most of what was discussed, so you can just continue. Don't wait for me. I'll just introduce myself. I'm Willem van Schaik. I'm an associate professor at the Department of Medical Microbiology of the University Medical Center in Utrecht in the Netherlands.
Yes, thank you very much for joining us. We're glad that you made it. Do you have, now that we've got you, I'm just going to put you immediately on the spot because we were just talking about the accuracy of assigning these short sequence fragments. And I know that you were specifically looking at, there were a couple of points that I noticed that you made, and if you don't mind me putting you on the spot, I might ask you if you wanted to elaborate on them. Like one of the first things I saw you mention on Twitter was to point out the, I think it was the 2 insects that they had noted, the Mediterranean fruit fly and the mountain pine beetle. And you had kind of pointed out like that it's very, very unlikely that those are the true identifications for those sequence reads.
Yeah. I've been fairly critical of this paper on Twitter and also in the real world. And one of the problems that I saw was that so they use short sequence reads to map to databases and to certain genomes, and I guess that's all fine, as long as you don't, as long as you take care of what you're doing. And that I'm not so convinced. And one of the things that I found when I went through the supplementary information of this paper is that they map lots of their reads to, I can't remember the exact name, but some kind of fruit pest that's actually not known to be present in the US outside of Hawaii, if I'm not mistaken. So obviously there's something going wrong in that analysis at that point.
I think it's not just that it's not present, it's that if it is present, it's a major problem.
Oh yeah, no. If they should close down the New York subway, not because of Yersinia pestis, but because of this fly, probably.
And that's because, I'm sorry to interrupt you, but when you mentioned that, I went and looked into it because I know nothing about insects, even though I've worked with ants. I worked on the bacteria, but I went and looked it up, and I thought, oh, so this is it's a pest that's being actively monitored by the United States government to make sure that it doesn't make it into the country. Is that right?
Yeah, exactly.
So the fact that it showed up, I think, is notable because that's very unexpected. So people are monitoring to make sure this doesn't show up, and then through a sequence analysis, it appears that those reads are from that, but it's a very minimal amount of information to make that conclusion.
Exactly. Yeah.
And you also, I think, unless I'm misremembering, I think you also you were talking a little bit about Yersinia pestis. I think, is that true?
Yeah. I think that was my sort of major criticism on this paper because, well, they make a claim of, well, they claim that DNA of Yersinia pestis and B anthracis is present on the New York subway. Mm-hmm. And actually, there's a major error in one of the figures, in figure 3 of that paper, where they claim to show that they have reads mapping to YMT, which is probably the main Yersinia pestis virulence factor. Mm-hmm. But if you look carefully at that figure, actually they don't show the so they show the virulence plasmid of Yersinia pestis, and then they have sort of a blowout, which shows reads mapping to a certain region of the plasmid. And actually the region that they show is not the region which has the YMT gene, so the Yersinia pestis virulence gene. And Chris Mason has sort of confirmed that, oh yeah, they made a mistake, and he showed in his blog post on, I think it was microbe.net. Mm-hmm. Yeah, okay. He showed that basically he showed a sort of complete mapping of all kinds of reads, of all the reads to the entire plasmid. And basically there's large parts of the plasmid where reads are mapping to, but these are actually parts of plasmids that are found in other gram-negative bacteria. So they're in no way indicative that Yersinia pestis is present on the New York subway. And actually he cannot show that there is reads mapping to YMT. So in my opinion, there's absolutely no reason to believe that they found Yersinia pestis DNA on the New York subway.
So I think the other, taking the larger context too, so there's a lot of Yersinia genomes. We know a lot about phylogenetics, phylogenomics of it. We sequence the ancient genomes, right? And there's actually no visible SNPs between the ancient genomes and the ones that are present, right? And so to find 700 SNPs in a couple kb region is a little bit of an outlier versus what's known in the context of the variants in Yersinia too. So
exactly, and it's sort of the same story, I suppose, with the Bacillus anthracis data, although they don't really show it in the paper, and I haven't got through that supplementary information that in great detail to check for let's say the evidence for B anthracis, but I assume that they're mapping reads to plasmids of B anthracis, the virulence plasmids of B anthracis, that can be found in other closely related bacilli like B. cereus and B. Bacillus thuringiensis. And I would be maybe sort of convinced, or it would be more convincing if they would show reads mapping to the toxin genes or the capsule genes of Bacillus anthracis, but I did not find that in the paper. And I'm very highly skeptical of actually them having shown even traces of B. anthracis DNA on the New York subway.
So do you go ahead, Dave? I'm sorry.
Larger point, and you'll see that. I think you'll see this problem showing up as data becomes bigger, and the studies become more known. Right? There's a lot of, when you talk to the specialists that really like it, I know Pseudomonas genomes, right? I know what to look for. I know it's there. I know kind of what's characteristic of what I expect of Pseudomonas genomes. When you get these big data papers that are just sampling what's there, taking the 30,000-foot view, like you can say, oh, this read maps to this genome, this read maps somewhere else. But unless you're used to looking at the genomes and knowing what to look for, it gets hard to really interpret that data at a fine level that I think they tried to write in the paper, but they didn't quite. That was where the problems really arose. And I don't know that there's any way around that because we're just sampling everything now. And it takes time to actually learn what to look for. So I don't know that there's a way around it, but I think it's going to be a larger problem going forward in the future.
Yeah, and that kind of gets to a question that I wanted to ask about this, because I think that, I think this is a valuable thing to study and an important question to ask if you're looking at, we just lost Willem, that's funny. This is a valuable question to ask to understand, as people are moving through the subway, as people are coming into contact with surfaces, how are the microbes being deposited? Are they colonizing? What happens? And I don't know, they talked a bit about trying to use culture techniques, but as we all know, that has its own huge limitations in terms of being able to recover accurately the population of microbes from a surface on a plate. It's the great plate count anomaly, which some people think we're missing 99% of what's there. So given that we have these limitations, it makes sense that people are trying to use these short read sequencing tools to do this. I'm wondering, though, until we can get longer reads or better databases that either have more sequence coverage or better annotated sequence coverage, what do you do? Is it that you should cut yourself off at a certain level of classification? Are you is it dangerous to say, like, I'm going to try to classify this down to the species level?
Well, I think, and Chris Mason's post on the MicrobeNet, it kind of gets to this point, and there's artistry in writing papers, right? And he acknowledges that there's a lot of slop and a lot of interpretation and nuance in his data, but then the paper overwrites that, right? And so I think this paper would have helped if there was a paragraph saying, look, this is what the reads look like, this is why we should interpret it not that way. But they don't write that, and I'm not sure if that got written, got cut out, I'm not sure what happened, but in his blog post he acknowledges that. And so I think in papers like this, it's especially with saying there's plague or bacillus or anthrax in the New York subway system, those kind of nuanced paragraphs are necessary to kind of place things in the right context.
Hey, Laura?
Yeah?
I don't mean to cut you off, David. We're having all this wonderful discussion of other sources of information that we can use to analyze this paper. Could you put those up on the website, maybe, so folks can have a look at that?
Do you mean like the blog post that people are talking about? Yeah, I meant to get that up earlier, and I didn't have a chance to do that. I'll definitely post that to the Journal Club's website. Because
my guess is, after hearing conversations like this, people are going to want to see those, even if they haven't seen them from before. This is a real value to this. This is exactly how to analyze research. This is a really good thing for students to see, if nothing else, let alone other scientists.
Oh, sure. I think that because these studies are a valuable tool, but I think, and I wrote a small grant to do a microbiome project, but I'm very conscious of the pitfalls, so I'm always trying to kind of keep track of studies like this that come out and what to pay attention to. Juan, you said that you're working on some metagenome studies. How do you see this where do you see this going? If this particular study was repeated in 2 years, do you think it would vastly change?
So I think it all depends. Well, the type of metagenomics we were working on was on hypersaline environment. So basically, we were looking at it was some places where we didn't have a lot of reference genomes to compare with. So we were doing a lot of assembly-based metagenomics. So, for example, in my opinion, it's something that I would love to see. It's more assemble-based analysis, but that's some kind of limitation where you're looking only at short reads, because then it's very difficult to try to sample something out of that. But I hope, and I think that will happen in 2 more years, for example, if something like this will go and happen again, is that reads will get longer, it will get slightly easier, maybe at least from the sequence part of the analysis, just try to start doing some assembly, or maybe trying to get some gene fragments, and not only trying to look at short reads and trying to map those reads to a reference genome, and trying to identify novel genes, novel organisms based on that from there. So, and that's something I was looking at this data, and I was wondering, for example, why they didn't try to assemble. But then, yeah, you see, it's a lot of sequence for each of the samples, but then you have a lot of diversity there. So trying to recover something from there, it's probably impossible, and it's a lot of sequence and very short reads. So I think that's a limitation on this particular type of study, and hopefully that will change over time.
Can I add to this? So I know that. So Ed Young asked me. So what for? Well, let's put it differently. So Chris, in his blog post, said something like, "Yeah, well, we have a problem with short reads, and we need to map to reference genomes, and that gives these spurious results." I don't fully agree with that, actually. I think that if he would have shown reads mapping to the entire Yersinia pestis genome and plasmids, I would have been quite convinced that there was Yersinia pestis DNA, and not live bacteria, that's a different thing, but at least Yersinia pestis DNA on the New York subway. So I think that the technology that we have now already allows us to do sort of fairly in-depth metagenomic studies based on read mapping. But I'm not sure how you all feel about that.
Can I add something to that? I think that's a very important point, but that, and I think it was what David was mentioning and Mark was mentioning before, I think that requires also that you have a question beforehand. So maybe say like, okay, I'm going to look for these particular toxins or this particular genome. But when you're looking at data or at all the data at the same time, probably you're not thinking that, and yeah, and I think that's what requires maybe that additional step of like, oh, okay, let's go and let's take a look at these particular genomes and let's try to map these genes and find the toxins and.
But I think something where we have 16S. That's the standard for a reason. So as we go into metagenomic studies, maybe we have to have standards. What's the standard for Yersinia pestis, right? When we map metagenomic reads, what's the standard for actually seeing that? What's the standard for anthrax? Things like that, where you can actually have something where you have an informed hypothesis about if Bacillus anthrax is really there, what should be there, versus just kind of let's map everything and see what the best hit is.
Okay, so that I like that. So that's almost, you've got, if you're going to collect all of this data, you can have different pipelines depending on your question. So, I mean, at some level, you kind of have to step back and look at the whole forest. But then if you're going to pick a tree out and say, oh, I found an oak or something, you're going to need to have some very specific criteria for why you're going to say that specific species is definitely there. Yeah, and Willem, I like your point about the read mapping. I've made a note about that when I was reading the paper, just to say, couldn't you, if you are going to pick out a tree from the forest, why wouldn't you just be able to take the reference genome that exists and map everything to it, and just like you said, say, here are the number of bases that had no coverage. 90% of the reference genome had coverage over 20x in this sample, so this is more convincing that we think these are the genes that we know are present. They probably indicate that this whole genome is present. Yeah, that's a good point.
Well, I know you're going to get to this later, but the thing that worries me about it, it takes me back to my criticism of some approaches as being swab stories instead of sob stories. In that, lo, I found in this particular surface a pathogen. And I really do believe that we're immersed in a cloud of microbes all the time, and I actually don't know the answer. If I do a swab of surface skin of humans, how often do I find MRSA? My guess is I do find it commonly, but it's an ecological question. How frequently and what's the balance with it? So I wouldn't necessarily worry about finding a pathogen, in a subway system. The real issue is how prevalent it is, and I don't know that they can address that very clearly. Perhaps I'm wrong about that.
So the MRSA was actually a place where they looked at MEX-A, which is kind of one of the diagnostic genes for MRSA, right? So that's where they actually went in and did more in depth, and I believe that there's probably a lot of MRSA out there, and that's...
No, but actually they make a mistake there as well. So actually lots of coagulase negative staphylococci, like Staph epidermidis, also carry MEK-A. So they become methicillin resistant, and it is believed that these staphylococci are the source of MEK-A that is taken up by Staph aureus, which then becomes methicillin resistant Staph aureus, so MRSA, or MRSA, as you say, on the other side of the Atlantic. So they show mapping to MEK-A, so the methicillin resistance genes, but I'm not convinced that they actually show presence of MRSA. They just show MEK-A is present, but MEK-A is present in lots of staphylococci.
That's a good point.
And I would argue that actually the bacteria that we're now talking about, so Bacillus anthracis, Yersinia pestis, and staphylococci, are incredibly easy to culture. And you don't need to do metagenomics to detect these three bacteria if they're viable. But
so that kind of brings me to a question. So Mark asked about prevalence, which I think it'd be really hard to judge. There are so many variables in this process, just in terms of your success at recovering DNA from a sample. I don't think that your read depth would necessarily always correlate to prevalence in on a railing, let's say. Like if you swabbed a railing and then you went through this entire process of extracting DNA, it's Illumina, so it goes through an amplification cycle in the process of sequencing, and then you are analyzing it. I don't know that you could yet tie sequence read depth coverage to prevalence in an environment. I'm not confident that that's connected.
So DNA purification, again, it's going to matter for, if you got gram positives, they're going to lyse a lot differently than gram negatives. And so I didn't dig into the methods to see if they added lysozyme or stuff like that, or if the mobile kits cover it. But that's another, amplification, you've got extraction, there's all different types of biases that kind of, it makes it difficult to relatively know, or to normalize to who's actually there or not.
Yeah. So my other question, and I honestly don't really know the answer to this. When you're doing these studies where you're recovering DNA, the point was made about whether or not they're. I mean, Willem mentioned, are they even viable if they're there? If you can culture them onto a plate, then you have viable cells. But if you're just going off of read data, I honestly don't know. Does anybody know what the relationship is, or has there been any kind of evidence of how often you're recovering DNA from cells that really would not be considered viable anymore?
That's another point he makes in the blog post. They needed to do better controls to figure out how long DNA lasts around out there.
Well, and they mentioned that in the method, or somewhere in the paper. Oh yes, it's in the discussion, and I thought this was really interesting. They mentioned additional positive controls are sorely needed for future sampling protocols. This could include barcoded, synthetic, and titrated oligonucleotides being sprayed at regular intervals to account for the degradation, disturbance, and dissemination of DNA. That's interesting. I read that and I thought, ooh boy, wait until somebody gets a hold of that in the media. They're spraying DNA in our subways. But that's an interesting idea, because I don't know if we know what to expect with that.
I think what some people have tried to do is to analyze RNA, so to isolate RNA, and then to detect 16S or whatever. I don't think anybody's ever tried to do well, yeah, of course, people have done that, metatranscriptomics. So assume, let's say the assumption then is that RNA is far less stable than DNA. So if you can detect RNA of a certain organism, it's much more likely to be alive. I think it still will not prove that something is alive, but it's at least it's stronger.
Oh, drat. I think that was before he got rudely dropped from the call. I think that was a good point that he was making about looking to RNA to try to kind of understand the dynamics of how DNA persists. Yeah, so I think we kind of started talking about the methodology, and then we've spent a good part of time kind of digging into that aspect of it. And I know we're kind of coming up on 3 o'clock, even though we got started a little late. I don't want to keep us more than an hour. But I did want to mention one of the things that I thought was really interesting about this type of a study was the fact that you can, I mean, there's always the risk of you're kind of going fishing, which is very reasonable because you're attempting, as people who've talked about the research in microbiology of the built environment mention, it's important to try to get a baseline to understand if there's perturbations or variations. How do you even judge that? So it's good to start building this data. But one of the things that I think they kind of stumbled into that turned out to be a really interesting part of this paper was their discovery about the subway station that was flooded during Hurricane Sandy. And to me, I thought that was one of the kind of striking things that popped out that said, oh, wow, I'm sure maybe they did have a hypothesis that they would find that. But, I mean, that turned out to me to be a good point to argue about why we would want to do this type of monitoring, this type of study, because you end up with these naturally occurring events that you can then find these interesting microbial signatures as a result.
You're blowing over the biggest result in the paper, which is that my study organism of choice, Pseudomonas stutzeri, is everywhere.
I was going to ask you about that. So stutzeri, yeah?
Do you think it's contamination, David?
No, it really is everywhere. It's a pretty hardy industrial.
Wait a minute, David. Were you in New York? Were you taking the subway? Were you waving files around?
I grew up in the subways in New York, so
there
it's all. No, I think getting back to the Sandy point, though, I think it's a really interesting. It's an interesting result for a couple of reasons. One, you can see the echo. Two, you can actually, as this subway system is being, the station I think is gonna be opened up again, and so you can start sampling and see how long it takes to turn over to kind of the normal subway system that's there. So it's a nice jump-off point for a lot of cool studies that can happen in the future too, I think.
So last semester I was teaching freshmen about issues of symbioses and parasitism, and one of the things we talked about is this idea that some of Jack Gilbert's friends have pushed about a microbial aura. You touch, you're in an area, it kind of takes on characteristics of your microbes, you leave, kind of falls away, etc. And many of my students asked about competitors. What if two different people, are they battling it out for supremacy? And that's really a really good issue. I've often said if you think about microbes coming into an area, there's this just passing through versus squatters that are there shortly versus commensals that set up shop. And that's why the Superstorm results are interesting. It suggests there are niches that can be exploited even in places that you might not think are really amenable to that.
Yeah, definitely. So Karen, I'm going to single you out because I don't think they found any Francisella, which probably they would have included in their Yersinia and Bacillus. Would you expect them to find Francisella?
Well, my thoughts on all the finding, aside from all the technical difficulties that we've just covered, is kind of like what Mark was talking about in terms of the aura. So, I mean, anthrax and plague might have been in the subway at one point in time. So I'm just wondering if, like, aside from it, let's hypothesize and say that aside from all the technical difficulties that they did find it, how long has it been there? Like, how far is, because we don't understand what the turnover is on surfaces. So it might have been there at some point in time. Maybe it's not there right now, but how far in the distant past was it there? That that was what was more interesting for me, is the sense that you have microbes that are in the environment and you can detect them in time points. And even when they were talking about in the paper, in that figure, when they mention that peaks of people traveling in and out, that you have alterations of the detections of species diversity and amount of specific species, it led me to wonder: our microbiome is fluctuating on a daily basis, but does that mean that those microbes are also fluctuating on the surfaces as well? But those are my thoughts on when I thought the interesting points and when I looked at the paper.
Yeah, and I think that's, I mean, ultimately one of the arguments that they make for why you would want to do this work with the New York City subway is they kind of invoke the sense of pathogen detection, disease outbreak surveillance, bioterrorism surveillance, and I think it's the point that you just made, Karen. It's not only that you would just take these samples and have a baseline, but you do also need to understand variability. You know, what would you be looking for to be a signature that is going to set off some alarm bells for you? And is it just that, you know, as we've just talked about, there's the potential for mistakes or errors in how reads are assigned. So what would it take you to say, we have a bioterrorism event, or we have an outbreak event, and we can track it to concentrated from this station or that station?
So biological context matters a lot here too. So if you have anthrax spores, that's a lot different than Yersinia. Yersinia is really hard to get, just if it's on the surface, right? You have to have like an open wound, and there has to be enough there to get in. But if it's in fleas, then that's a different story. So there's, you know, just seeing Yersinia there doesn't mean it's really that transmissible, even if, you know, in the real world where it was there. Seeing the DNA or seeing bugs living on surfaces doesn't mean that we should freak out over, like, I live in the southwest of America, right? There's Yersinia everywhere here. There's like 2 plague cases a year. So
yeah, I told my class recently that there is still plague, and they were like, "What, really?" And they're like, "Yep, there's still plague."
So the other thing I just want to point out before we leave, and this, you know, we've talked a lot about, and, you know, journal clubs are kind of critical by definition, but this paper, it's really, there's a lot of information that they bring in, and there's a nice interactive map for where they sampled, and all the GIS data is out there. So this really is kind of a nice platform for doing these kinds of studies in the future, where you have multi-dimensional data where it's really cataloged and correctly. And, you know, they may miss on the specifics, but that's not to take away from the fact that this is a really cool data set to play around with too.
Yeah, I think that's
I agree. Yeah. This is in some ways a very, very cool paper, and it's a shame that almost the conclusions are a bit iffy, and that particularly these conclusions, have been picked up by the media.
Oh yeah, that's a big deal with the media on this subject. This whole microbial literacy that we try and promote, and I try and discuss this with lots of people, and they get the sound bits, the sound bites, and that's all they run away with. And that's why, actually, I love the bit, and I don't know if anyone ran Platypus Conquistador through this, but I love the Rob Knight story about that, and I'm hoping folks use that as kind of a way of, you know, kind of suggesting to take some things with a grain of salt.
I found a few reads mapping to crocodile and alligator in that data set.
Hey, but
so
you don't know,
clearly,
they could be in the subway. Somebody had a pet, and then it's just down there. That's why you don't mess with that.
That's a famous urban legend, right?
Yes.
Yeah.
Yeah.
So we're kind of coming to the end of our hour, given our difficulties. So Dave, you made a really great point about how this integrates a lot of information. I just wanted to give an opportunity to you to kind of, anybody who wants to have some final thoughts about the paper or the conversation today. It looks like Juan had something to say, and then nothing happened. Okay.
My all funding for should go to Pseudomonas stutzeri.
Yeah, you get to cite this paper now in all your grants.
But David, can I ask you just one more question about that particular finding, do you believe it's plausible that 50% of their reads map to Pseudomonas statzeri? Or do you think it might be?
I bet a lot of the reads probably map to things that are present in a lot of Pseudomonads. And so there's not, from statzeri to aeruginosa to syringa, there's a lot of Pseudomonads out there where, you know, there may be at a DNA level kind of 50, 60, 70% similar. It's like how it's not necessarily mapping to statzeri, although that's probably out there a lot. Like you get that in culture studies a lot. But there's definitely a lot of Pseudomonads out there.
Yeah, yeah. I know. Nick Lohman believes most of it might be contamination. That they find so much mapping to one particular organism, and he believes it might be contamination. I basically don't know. They could have solved it by doing a negative control on their kits, but they didn't. That's Nick Lohman finds that a major flaw in the paper.
Well, I think Dave, what you need to do is do fish mapping. And find yourself something specific. Get yourself a section of a handrail, and just dye that sucker and see if it's actually there to that number. And I'm only half kidding.
That's all I need is to go and spray. I'm spraying nucleotides in the environment then, fluorescent ones nonetheless, right?
Yeah, glow in the dark. Must be radioactive.
I think you'll be visited by some interested parties of law enforcement if you tried to do that. Okay, well, we'll wrap up here. I wanted to thank everybody for struggling through our Hangouts technical difficulties in order to join us today. I'm really thrilled that we had such a great turnout of people. I'm glad that we got a chance to talk about this paper because it did, when it came out, I saw a lot of discussion about it, and I think it was maybe Karen and Dave's suggestion to cover this paper, and I think it was a very good one. Thank you everybody for joining us. I really appreciate it. And this will go up on YouTube for posterity. I didn't tell Willem that because he joined us late, but I think everything you said was quite well supported, and I think it's, as Mark said, a really good resource for interested parties, including students, to kind of hear some discussion about the ins and outs of this particular paper. So thank you very much, everybody. It's probably time for bed for Willem. I don't know. What time is it there? I don't know the time zone difference.
9:15 p.m. So
9:15,
no,
okay.
not time for bed yet.
We're not keeping you too late. Yeah, thanks very much for joining us, given
okay.
and your technical difficulties. So.
Yeah, sorry about that.
Oh no, that's fine. So thanks everybody, and hopefully we'll in a month or so we'll probably come together again. And if you are able to and would like to join us, that would be wonderful.
Great. Thanks for organizing, Lauren.
Indeed. Thanks to both of you.
Yeah.
Okay. Bye. Bye.